So many of the themes that we've already heard about, I'll be building on as we go through the talk. But my remit today is to try and speak about potential future developments in the management of neurosarcoidosis. This image at the top right is very definitely not me with my crystal ball, but I hope that by the end you will understand that we do have a plan and a structure to deal with some of the uncertainties and the gaps in our knowledge that we've already heard about today. So first of all, I'm going to start by describing why neurosarcoidosis is a difficult condition for researchers. And I put that in inverted commas. It's obviously a very difficult condition for many people to live with as well, but it has very specific problems and challenges that it has for researchers trying to improve treatments and outcomes for patients. But I'm going to try and give an overview of what's been done to improve our understanding of the causes of neurosarcoidosis, understand the diagnosis, how to make that earlier and more accurate, and how to improve treatments and make sure that they're available for patients when they need them. Along the way, I'm going to highlight a few techniques that I think are worthy of special mention, and I'm going to try and highlight what we as a group of UK neurologists with specific interests in neurosarcoidosis are trying to do to advance treatments and diagnosis in the here and now. So why is neurosarcoidosis a particularly challenging condition for researchers? And this slide is really the equivalent of getting your excuses in early. It's trying to explain why we're not further on with our understanding of the disease, despite a lot of research that has already gone before. Well, as many of the speakers have said, neurosarcoidosis is a relatively rare condition, and it does present in very many different ways. For clinicians who rely on pattern recognition to make a diagnosis, that makes it more difficult. It's relatively difficult to study tissue in neurosarcoidosis. Obviously, nerves, the brain, the spinal cord, it's very precious tissue that can't easily be sampled. And we don't have much in the way of redundancy of that tissue. So all of it is precious, and taking samples of it to look at under the microscope or perform other tests on can be difficult because it can leave patients with symptoms afterwards due to the procedure, and the procedure itself may be risky. There are definite variabilities in terms of how patients respond to treatment. So as Dr. Aaron already explained, we've got lots of different treatments, but we do have difficulty predicting sometimes which patients are going to respond to which agents. And that, again, makes it difficult. Clinical trials are easiest when all of our patients have a equal severity of disease, and they present in predictable ways. That way we can monitor responses to treatments and measure those accurately. When patients have mixed disabilities, mixed symptoms, and respond differently to treatments, that makes the clinical trials very challenging because it makes those measurements and the statistics involved to decide whether a treatment is effective or not very difficult. And finally, we also have problems within the nervous system in terms of how the immune system works there. It's sealed off from the rest of the body, the rest of the systemic immune system, and it can be difficult for drugs to access into the spinal fluid or into the brain and around the spinal cord, for instance. So what can we do? Do we stand around, rub our hands together, throw our hands up in the air? No, we need an absolute plan to try and take this further. So in the here and now, our diagnosis relies on an index of suspicion. This is a rare condition. Clinicians need to be aware that it's a possibility. They need to be aware of the different myriad ways in which it can present. And we all have a role in education and raising awareness to improve that index of suspicion amongst the people that neurosarcoidosis may present to. As we've already heard, this is a condition that can involve nearly any organ of the body, and therefore nearly any hospital specialist or GP could be presented with a symptom related to neurosarcoidosis. So we have a job to do to get that message out and make sure that all clinicians and medical students are aware of this condition and how it may present. I've talked about how good clinical trials are done best with large numbers of patients with predictable disease and standardized disease. And of course, that is a challenge in neurosarcoidosis. To get over that, we're going to have to cooperate nationally and internationally to build expertise and design good ethical research that has robust outcomes. In order to try and strategize our approach to this, we have to think about exactly what we're trying to accomplish. Clearly, we want improved diagnosis and treatment for patients so that we can optimize their outcomes. That's relatively straightforward, or at least non-contentious. How will we know if a change actually is an improvement? I think it's very important that we listen to patients when trying to design our outcome measures in this regard, and we do have a range of patient-reported outcomes in order to help us do this. Added to that, we have clinical scales, so measurements of disability, for example, that will help us determine whether something that we've done helps or doesn't help. And then in addition to that, we'll have test results, so blood results, scans, etc. And we'll come back to that in a little bit more detail in a bit. And then finally, we need to know that we have some kind of intervention that we think has a good chance of success. That might be lifestyle changes, drug-based therapies, or other advanced cell-based therapies, for instance. And again, we'll talk through some of those. But all of these three different parameters require an improved understanding of the disease process if we're going to optimize the benefit observed. So how do we look into the cause in a structured and sensible way? Well, right at the beginning, we talked about some of the studies that have already been done to understand how the disease affects people in terms of which people it affects, what part of the world they live in, what race they are, any of the risk factors that might predispose them to developing neurosarcoidosis. This type of research is extremely important, but it relies on huge data sets and good quality analysis of the data collected. Basic laboratory research has a lot to offer, and there have been enormous advances here in the last 10 years in particular, as other speakers have alluded to. Back in the old days, someone could have taken a sample of tissue, brought it back to their own laboratory, such as we have right next to our clinics, and run some tests and gain some useful insights. But those days are gone now. We are working in an advanced scientific field, relying on collaboration with specialist scientists working in multiple different disciplines. Genetics, for instance, has a particular role to play, where we can look at susceptibility genes to try and determine which patients are more likely to be affected by the disease and use this to highlight cell pathways of interest. Those are pathways by which cells might communicate or by which their metabolism and immune system works, so that we can interfere with those to reduce the severity and likelihood of disease. We can look at cell populations in increasingly sophisticated ways. So not just B and T cells, cells within the immune system, but much more detailed subpopulation of those immune cells. Some of you will already be familiar with rituximab, which we talked about a little bit earlier today, which is an anti-CD20 therapy. So picking out those particular cells, expressing that flag on their surface and removing them from the circulation. Identifying the different flags that cells of different subpopulations display will help us generate new and ever more targeted therapies for a range of different conditions, including neurosarcoidosis. And the science will also help us understand how cells communicate or signal to each other. An example of this is already in clinical use, as we've heard. Infliximab targets the tumor necrosis factor alpha, which is a cell signaling mechanism. And by reducing that, we know that it has many beneficial effects in neurosarcoidosis. Again, Dr. Aaron mentioned the JAK inhibitors and STAT2 pathways. These can be, again, manipulated to reduce the likelihood of disease, or so we hope. In order to test these different theories that have been generated by the basic laboratory research methods, we need disease models. And these range in complexity from whether a specific compound can increase or decrease the activity of a cell in the laboratory itself, to more sophisticated models where we can actually try and generate the granulomas that are the hallmark of sarcoidosis and examine whether drugs and other compounds may reduce the likelihood of a granuloma forming, or look at how the cells move in response to that compound or drug. Does it make it more likely that immune cells will enter the central nervous system, for example? In some diseases, we have good animal models in which we can test drugs and other interventions before we try testing them in patients. But unfortunately for neurosarcoidosis, there is no widely accepted animal model available. at present. This again is another area in which there is active research. I'm just going to highlight the power of omics as a special candidate highlighted in this slide. It's a term you might have come across before. Omics refers to the study of the different building blocks that goes all the way from our genetic code, the DNA itself, and how that is translated or interpreted to make the proteins and the other tissues that make up our blood and tissues such as the organs in the lung and central nervous system. And on top of this, there are the things that we put into our body or are affected by our environment. This last one is called the metabolome. Each stage of this process, going from the genetic code itself to building up the different proteins and how they interact with our environment, the study of this generates enormous amounts of detail. Billions of data points can be identified from individual patients. And putting this data together is a real logistical challenge, but it also does have incredible power to tell us detail about the disease that we've simply never had before. Clearly, all of this is expensive, and it also requires the skills of an inordinate number of different scientists rather than individual clinicians. But this really is exciting for those of us who want to understand complex diseases, where we know we have gaps in our knowledge about what causes them and how best to treat them. So this is an image that's hot off the press from October of this year, so next month's edition of a Nature Reviews paper. And it's a complicated slide, but essentially it's pointing out that all of the information and data points that we can glean studying that process from the genetic code all the way to the construction of proteins and how they interact in our bodies with the environment has enormous application in terms of discovery of different cell types relevant to disease that we simply didn't know were there or missed them as subpopulations of larger ones, to understand why the tissues respond in the way that they do, to understand why cells are present in different parts of the body or attracted to different parts of the body to cause disease in one patient rather than another. It can help tell us about biomarkers, things that we can pick up in blood or saliva or urine or spinal fluid to try and help us predict who's at risk of disease or how their disease is responding to treatment. And it can give us enormous insight into complex diseases where there simply isn't one cause, simply as very much as we suspect in neurosarcoidosis. And finally, by putting all that together to understand the pathways and networks of cells and proteins, we'll be able to identify novel treatments. No talk on the future developments of diagnostics and treatment would be complete without mentioning imaging. We've heard already how much MRI has contributed to neurosarcoidosis and sarcoidosis in general, and further developments are being made all the time, in particular with respect to high field MRI. So essentially, the more powerful your magnet, the more detailed the image that you can generate. Functional imaging, looking at how blood flow changes in parts of the brain that may be susceptible to inflammation, for instance, and also looking at vessel wall imaging. The image at the bottom of the slide, labeled C, shows with the arrows a vessel that's been affected by vasculitis in neurosarcoidosis. And this level of detail has simply not been possible previously without injecting toxic and potentially damaging dyes to visualize this. Nanoparticles as novel contrast agents are being developed. This is in place of gadolinium. So gadolinium, as many of you will know and have experienced, is useful in highlighting areas of inflammation within the brain, but it isn't specific to neurosarcoidosis, and some inflammation can be missed. So nanoparticles are being developed to home to these areas of the brain and spinal cord where there's increased activity due to inflammation. Similarly, in trying to predict or highlight the areas of brain and spinal cord that can be affected, new PET ligands are being identified. So many people will have had a PET scan, which essentially shows, again, cell activity. So it's a radionucleotide ligand patched onto some glucose. And where the glucose is taken up is where the cells are particularly active. And this is highlighted on a CT scan. But it doesn't, again, it isn't specific, to particular types of inflammation. And other reasons for cells being active, such as cancers or malignancies, may also be highlighted. We're interested in specifically diagnosing neurosarcoidosis, and therefore we want to track very specific subpopulations of cells with different flags on their surface. These are the new and novel PET ligands, which we hope will be identified in future to try and make the diagnosis more quickly and more accurately. And the image on the right shows some of the potential options for this, again, targeting various different specific cells. It's likely that by putting all of these different techniques together, we'll be able to optimize our diagnostic processes and processes for monitoring disease activity. And the cardiologists have really taken the lead in this. And it's now very common in cardiac sarcoidosis to have a range of different tests looking at inflammation in and around the heart, not only to diagnose sarcoidosis, but to see how effectively it's being treated. I've talked about how much data we're going to be inundated with as these new techniques come to fruition. And artificial intelligence is very much in the news at the moment and has undoubted potential application for complex diseases such as sarcoidosis. It's not going to be possible for a single clinician or a single scientist to put all of this data together and get the most out of it in terms of diagnosis and predicting response to future treatments. We will need the help of complicated algorithms and specialists to help us design these programs, which will be able to be most predictive and most accurate in future. I think this will generate new information about potential new treatment options, but I think also importantly will enable us to compress what is a very long process of taking potential compounds that may be useful as future drug therapies for a condition and putting them through the clinical trial process, which can take decades in some cases. With artificial intelligence and screening, putting all of this information together, it's hoped that we'll be able to compress this and allow us to get new and exciting drugs to patients within a much shorter period of time. So to date, we focus on our understanding of how to improve our understanding of neurosarcoidosis. And any of these improvements will obviously have an effect on diagnosis. But I'd like to highlight particularly the potential for biomarker development and improved imaging techniques in terms of translating that into benefit for patients within a relatively short space of time. But if we're going to continue to improve our diagnostic processes, we do need to improve clinical training and expertise. And as Dr. Oren and others have mentioned already, we also need increased multidisciplinary and speciality team working. And increasingly, that won't just be clinicians and clinicians in other specialties. It won't just be working with radiologists, nurses, occupational health therapists. It'll also be working with bioinformaticians, looking at all the data points from individual patients and translating that into a predictive algorithm that will help us predict which treatments they will respond to best. So if we are going to improve treatment in the here and now, we need to start and make sure that we're doing what we should be doing at this point in time. I think we need to be sure that patients can get access to specialists and their MDTs. We need them to be able to have the options to get the tests they need and discuss the treatment options with someone who has relevant knowledge and practice in using those drugs. We need to balance the therapy. There are risks, as we have been highlighted already, of over and under treatment in neurosarcoidosis. We need to ensure that access to medications, including high-cost drugs, is appropriate. And we need to make sure that once patients are on treatment, they can be monitored safely and effectively, both for side effects of therapy and for relapse in disease activity. So we are lucky to have processes that we can go through in order to get access to drugs such as the high-cost drugs such as infliximab, and we're very grateful to Dr. Kidd and Professor Scolding who worked so hard to ensure access to infliximab. But there are other drugs coming and more flexibility in prescribing that we need to work with these agencies on. Retrospective research studies concentrate on looking back. They look at the outcomes of people who've been treated in the past, and they tend to work on the data that essentially happens to be there. We need to collect data on treatment, on the treatment of patients over past years, and ensure that we've learned from that data how best to treat patients in the future. This is something that with good registry data, we can do straight away. Going forward and acquiring new data, we want to be more structured about that. We want to have patients in whom we've got well-characterized disease, so we actually know exactly what symptoms they presented with and exactly how they were treated and exactly how they responded, ideally over a long period of time. And ideally, with outcome measures that were highly relevant to the patient and to the clinicians looking after them. In order to do that, we need to plan and structure and coordinate that data collection amongst large numbers of neurologists working all over the UK. That requires some coordination and strategy. As we've talked about, we have very poor evidence base for neurosarcoidosis, and clinical trials are very much a priority. There are things we could do straight away in terms of examining the existing drugs head to head. Is infliximab better than cyclophosphamide? Is it better tolerated, less well tolerated? We don't actually have hard data to give facts and figures to patients that they need. We do want, of course, to develop ever better therapies, and that might mean developing new drugs, but also what we call repurposing old drugs. A good example of repurposing drugs is it comes from the MS world, where a drug that's licensed for the treatment of diabetes, metformin, is now being examined for an effect on multiple sclerosis. These trials are quicker and easier because the safety data is already available for those compounds, and that saves a lot of time when moving a treatment through to the patient. Novel compounds have to be extensively tested for safety before they can be introduced to the patient. But both novel and repurposed drug therapies need to have a strong rationale for effect, need to be tested in disease models, and have a strong rationale for moving on to being tested in patients based on an understanding of the underlying disease process. If we're dealing with clinical trials in neurosarcoidosis, we need to be sure that the design of those trials is efficient and robust. I've already talked that they are more complicated or likely to be more complicated because of the differences between patients and because we'll have to recruit patients over a wide area with lots of different clinicians nationally and likely internationally. That again requires strategy and cohesion in order to do that well. Worldwide and in the UK, unfortunately, we've had a traditional problem ensuring adequate diversity of inclusion in clinical trials amongst people from non-European backgrounds. And that needs to be a priority going forward if we're going to have true representation of the people who are affected by sarcoidosis and, of course, neurosarcoidosis. And then we need to be clever about the choice of target that we want to test in our clinical trials. The diagram on the right is a busy slide, and I don't want to go through it all, but the point that it's trying to make is that on the left, there's what we understand about the pathogenesis of sarcoidosis. So lots of different signaling pathways, lots of different triggers, lots of different points at which we can intervene before the granuloma is formed. And on the right of the slide are lots of different potential treatments that we could test in neurosarcoidosis. Clearly, this is complicated. Some of these drugs will be more likely than others to be beneficial in neurosarcoidosis as opposed to pulmonary sarcoidosis, for example. And we need careful coordination and workup to ensure that our efforts and the finances involved are well targeted to the drugs most likely to help. So some additional considerations around improving treatment that I wanted to highlight. It's not all about drugs, and some of it is about the difficulties of drugs, including getting drugs into the central nervous system, for example. Nanoparticles are being developed as drug delivery vehicles to help get drugs into the area that is most affected by the disease. That may be of particular relevance to neurosarcoidosis, amongst other neurological diseases. And then chimeric antigen receptor-based therapies is something that you may have heard referred to as CAR T therapy for patients with hematological malignancies, such as leukemia or lymphoma. These are complicated therapies where cells are engineered to recognize triggers on other cell populations that are causing disease. So the diagram on the right shows a B cell that's expressing a particular marker, and the long string-like figure at the bottom is really the entrapment mechanism that the second cell uses to snare that cell and then destroy it. If we know the particular flags on the particular subpopulations of cells that are problematic in neurosarcoidosis, the entrapment snare can be specifically designed and those cells generated to remove the cells of interest. This is very much something for the future, but is a technique that is increasingly being used in other malignancies and beginning to be explored in autoimmunity also. There are some further questions that we're going to need to address before we can optimize treatment, and that again, we've alluded to some of those as the other speakers have gone through their presentations. That includes how can we predict relapse more accurately, and related to that, who are the patients in whom it's safe to stop immunosuppression, and who do we need to continue that for, and for how long? We also need to think carefully about how to minimize steroid side effects, which cause a lot of problems in our patient cohorts. We have a lot to learn from the rheumatologists who are really focusing on this very much at the moment, and are designing clinical trials to tell us where to place the dose in terms of getting optimal response between side effects and response to disease. So putting all of this together, really what we want is a personalized medicine approach. What do I mean by that? Well, I mean integrating all the information that we have about the disease process, the pathogenesis that Dr. Fernandez spoke about at the beginning, together with all the new research that's coming through from the bioinformatics, and putting that together with person-level information. So what are their biomarkers? What are their imaging results? What is their genetic background? So that we can make decisions about therapy based on individual characteristics. At the moment, we can only say this drug works in this population. We can't promise an effect in that individual, and we want to get better at targeting that. Unfortunately, at the moment, the modeling performs poorly, but artificial intelligence really does have the potential to improve this. And we will have a further barrier in that current healthcare systems really aren't designed to deliver this kind of bioinformatic-driven medicine. So we have some work to do to prepare it and to make sure that we're ready to deliver these changes when they become available. So what are we doing at the moment as a group of interested neurologists within the UK? We're setting up formal networks, multidisciplinary team meetings to improve expertise and to improve the education of our colleagues and raise awareness of the disease. We're working on building our international links and collaborations, and we're working hard within the UK to improve access to to treatments that should be currently available. Some of our immediate aims are to establish a UK neurosarcoidosis registry, and we hope that you'll be hearing more about this in the months to come. We hope to work together to set out some neurosarcoidosis management guidelines that we can share amongst our colleagues and neurologists within the UK and beyond. And we want to increase research opportunities for those patients who wish to participate. We want to focus on patient-reported outcomes, so the things that are important to you, to look at studies, to look retrospectively at the data we already have, but probably more importantly going forward to standardize that data collection so that we can do good prospective studies. We want to be able to collect blood, spinal fluid, and tissue from patients willing to donate it so that we can do basic research on that and collaborate with our colleagues internationally. We want to work with our neuroradiology colleagues to improve access to imaging modalities that could be available in the here and now, but aren't always either available or there isn't the expertise to interpret them, such as vessel wall imaging, for instance. We want to work hard to try and define that risk stratification that Dr. Arun talked about. Who are the patients who need to be treated aggressively and early? Who is it safe to sit back, protect them from the toxic effects of our medication or potentially toxic effects of our medication, but we don't want to risk future disability? And can we get involved in the new treatment trials that are emerging, collaborating with colleagues internationally to get the right numbers, the right cohorts of patients who are willing to accept some risk as well as potential benefit, given that they're participating in a trial of a compound that hasn't previously been proven to work, but that has potential to be better than currently available therapies. And again, we need to keep a very close eye on what we do with our treatments in terms of side effects and how to minimize those for patients going forward. I think that was my last slide. I just wanted to thank those at the Neuroinflammatory Clinic in Bristol who've gone before me, both Professor Scolding and Dr. Joseph, to thank all the colleagues at the National Neurosarcoidosis MDT, which has been a privilege to be part of, and to Sarcoidosis for the invite today.